So again, I've been doing this for 15 years. It's very, it's nice doing this kind of work in the sense that at cocktail parties, if I was working on, I don't know, glycolysis in the liver, and somebody asked me, oh, what do you do? What kind of research do you do? And I said, oh, I work on glycolysis in the liver. It's really exciting. It would take nanoseconds for their eyes to glaze over. Well, when I talk about what I do, I say, I work on marijuana. It's like, really? That's so cool. And I guess I'm just really lazy at cocktail parties because it's so easy to talk about this work. I guess we're going to talk about you may not know this, but IU is actually a center for cannabinoid research. Who knew that? There are, some could argue that there are more key scientists doing work, and I won't necessarily count myself among them, doing cannabinoid-related work here in Bloomington, Indiana, as any other place on the planet. It's a real concentration of researchers here doing interesting work. So that's kind of the package about it. But as a consequence, this is why I'm still here. I was planning to leave after 2 years. In 6, I'm still here. So it's doing good work. So a little bit about the history of marijuana. And just kind of give you an overview of where we're going. The idea is, I'm not going to spend a lot of time talking about what we do. I'll try to link it in where possible, but really for the most part, this is kind of an introduction, kind of the kinds of questions I've been getting over the years, things that people want to know about marijuana. We kind of fill in a little bit. This goes maybe half an hour, and then we'll open it up for Q&A. I won't have all the answers, but I will have hopefully a lot of them. And then depending on how many unanswered questions we have, we can put that up on the website. But so, this has been with us for a long time, and it's possible. It's sort of the first evidence of fabric, actually, comes from Taiwan, and it's embedded into clay, and that is hemp. It's definitely hemp, and that's from 10,000 years ago. That has been with us for a long time, but we don't know, where there's no ink or what. The earliest reference is from 4,500 years ago. This admittedly comes from something that is in fact only 2,000 years old. We're not sure about this. But the data certainly start talking about it. There are references to this that are thousands of years. So this plant has been with us for a long time. You figure they're using it on Taiwan. I think it natively comes from the Himalayas, some of the southern Himalayas. So someone had to bring it in the first place across water. So this has been around for a long time. Some of the early observations are interesting. So this is an Arab text. I forget exactly, I think it's maybe the 10th century. That's the Maximo Al-Rawaiya, I guess on the pronunciation. Describes it as a snuff to deterge the brain. I don't know what that means, but I like it. Against dandruff, as a topical insecticide. I don't know if that works. That might actually work. I'll try that one. But also against wounds, and stomach pain, and inflammation. And what's interesting about this list is that a lot of this is correct. A lot of this is valid. These are modern observations. Pain, inflammation, I don't know what's the source. But these are things that are currently being researched. Some of them are actually being researched here at RU. So people have been experimenting, exploring this for a very long time. Just some basics. So this is something that many of you I'm sure can answer just as well as I. So what is the difference between marijuana and hashish? Anyone?
Strength.
I'm sorry?
Strength.
Strength? Yeah, it's certainly strength. Hashish tends to be stronger. The way it's prepared, any idea? I don't want you to give anything away. I mean, if you're shy, I can change it for you. But no, it's a different way of preparation. Basically, kind of grinding and getting hashish, it's stronger form. And then what about hemp? What is hemp?
Plant fibers.
Yes. So we talk about hemp as being the fibers, but it's another word for the plant. And so there are different plants. We've through genetic breeding, basically, over thousands of years, there are different plants that have been developed with different qualities. But we've taken this to the extreme in the last 30 years. It's extraordinary what people have done with these plants. But there are some plants that are more, the hemp plant fiber. But those are also not allowed to be grown in the U.S. because they're classified, maybe because they're worried people will grow both or whatever. But the hemp can also have a very low THC content, in which case, but it is not allowed to be grown here.
Because it has a low THC content, it often has a very high cannabidiol content. Yeah, thank you. I knew I was pronouncing that incorrectly.
That's totally fine.
But that matters because there's some things, including medical stuff, that specific cannabinoid does. That's unique. Unique to it.
Exactly.
And so if you want it in large amounts, you can sometimes find it in
We'll talk a little bit about the main ingredients of components, the main psychoactive and that's what I call psychoactive components of marijuana and hashish, and kind of what that, partly what selective breeding has done. So one question then is well, how does marijuana work? Because people smoke it, so why does it do anything? And for a long time, this was really an open question. Does it change the lipid properties of neurons? What's going on? People weren't really sure. And so but this all changed in the late 1980s, rather. Basically in 1990, cannabinoid receptors, so obviously from cannabis is the root, cannabinoid receptors, cannabinoid field of research is what I'm in. And particularly, the first one that was identified was CB1, the cannabinoid CB1 receptor. And this is a receptor that's dedicated to receiving a signal. It's part of a signaling system in the brain and the body. And there's another one called CB2. It's been thought of as kind of the immune receptor in the immune system. It has been a little less well studied, but is also particularly. These are what are called G protein coupled receptors. Without going into detail, it's a super family of receptors. They signal, basically it's a way of taking an outside signal outside of a cell and getting it to do something inside the cell through second messengers. And so this is part of a super family of about 1,000 different receptors. And so there are two main cannabinoid receptors. And there is this question of whether there might be other receptors. That's still an open question. We've actually done some work on that as well. Looking at the localization of cannabinoid CB1 receptors in the rat. This would be the cerebellum of the bat, forebrain over here. Everything that's kind of red or orange is CB1 receptors. It's everything. It's really all over this brain. So they are ubiquitous. They're also numerous. So they're among the most numerous receptors of their class, which is interesting. Must be important. And there are also ancient. I'll show you a graph. Basically, all of these species have cannabinoid receptors. Even dinosaurs had cannabinoid receptors. So how did they smoke? No, so this raises the question of why do we all have this? Why is this around? Why has this been around for 500,000 years? It must be important. It's also very well conserved. The structure is very well conserved across the species. The body. So the question is, well, okay, so you've got receptors. That's great. But surely the body doesn't make THC, right? It doesn't make marijuana. It makes something else. It's a signaling system. It's got receptors and it's got ligands. It's got its own cannabinoids. And I'll show you the structure in a minute. The two chief cannabinoids at this point are called 2-arachidylglycerol. Basically, it's a lipid chain. And they're lipids, which is unusual. So usually when you think of neurotransmission, when you think of signaling, you think of little packets, little vesicles with packets of information that's thrown out into the synapse. Lipids are different, right? So lipids want to stay in the membrane, but here they are. So you have a lot of evidence at this point that lipids are serving as signaling molecules. So these are lipid receptors, and that's very unusual. So they cross the blood-brain barrier, which has a significance for us. And then the actin receptors in the cell membrane. But this is interesting. This is as a note. This is why, if one takes a urine test for exogenous, for phytocannabinoids, it can stay in your system for a long time because they're lipids. They get into the cell membranes. They get into your fat cells, basically. So they stay in your system for a long, long time. And that's why urine tests. Because some things you just get very quickly, but cannabinoids stay in for a much longer time because they're lipids. Structurally, they don't look a whole lot like THC. This is delta-9 THC. It's got these 6-member rings here. Anandamide is an example, the twitch is very similar. It's just a long chain of carbons. But what they are is that they're both lipophilic. They both like to stay in the membrane. This makes it very flat, and it's fairly non-polar. So it just gets right in there, into the lipid membrane and stays there. So what you've got these components, right? You've got cannabinoids, so that's a signaling molecule. You've got receptors. What do they do? What's the point? What does the endogenous system do? And it's basically a feedback inhibitor, and it plays a lot of different roles. But the biggest role is the feedback inhibitor. And so just as an example, imagine you have two neurons, neuron A and neuron B. And neuron A is doing the firing, neuron B is getting the signal. What this allows neuron B to do is say, cool, we got the signal, right? So this is a presynaptic neuron A. Neuron A, the terminal, the release site, usually you have an action potential and a calcium coming in, releasing neurotransmitter, crossing, and signaling to neuron B. That's the usual pathway. Action potential, calcium, neurotransmitter release, signal. If you activate cannabinoids, what you end up getting is a reverse signal. It goes backward across the synapse, activates CD1 receptors, and that inhibits the calcium channels. You don't get this kind of a neurotransmitter response the next time. So the next time you get an action potential, it's blocked. And that's what cannabinoids do. They block neurotransmission. They do it maybe for tens of seconds, but there are other ways that they can signal as well. And so depending on the circuit, depending on how it's organized, you may have a very different effect. So it doesn't just depress you necessarily. It depends on what the circuit is, where it's acting. If it's acting on an inhibitory circuit, you may actually be getting a lifting of depression. So it just depends on how it's done. Affected systems, a lot of different affected systems, right? So appetite, munchies, right? Everyone's heard about the munchies. Nausea, definitely, it's actually, there's considerable therapeutic potential for nausea. Intraocular pressure was actually one of the first medical purposes that was identified. We're actually doing some work on intraocular pressure. Seizures. Movement, there's a lot of interest in movement. Mood, there's actually some evidence that a part of the runner's high may involve the cannabinoid system. But certainly mood, experience euphoria. The number pain is a big deal, and there are a lot of researchers looking into pain right now in terms of what cannabinoids can do. Memory, especially the extinction of memory, because you don't want to remember things forever, you want to also forget. And so cannabinoids seem to play a role in forgetting, which is also very important. So for things like post-traumatic stress disorder, people are very interested in cannabinoids and maybe playing a potential therapeutic role. And of course, the usual joke after this is, there are other things, but I forget what they are. But, I don't smoke, actually, I don't smoke. But what's interesting is, so you think, my God, these receptors are everywhere. They must be, and they're really important, they're really well preserved.
Just one more medical thing on what you were pointing out. The fact that it helps nausea and blots out some of the memory of the chemotherapy and increases appetite are three things that you can't find in almost any other, even in separate drugs, to work equally as well during chemotherapy treatment.
No, there's definitely a lot of interest in therapeutic uses. We'll kind of talk about some of the pitfalls, some of the advantages, some of the pitfalls of that. Because if anything, there's an embarrassment of riches in terms of potential targets. But so it is possible to engineer mice that do not have these receptors. They're called knockout mice. We actually have a case of these mice, but they live just fine. So they're not essential. These receptors are not essential to living, which is interesting. Again, so they are more prone to seizures later in life. They do a little bit differently on some tests of memory, things like that. But the differences are fairly subtle. It's not essential to life, like some people would disagree. So medical prospects. Here we're kind of getting into some of these issues. So the one problem has been the concentration, the dose that it requires to experience euphoria, which is considered a downside in our society, that's a fairly low dose. So you often have to go past that to get your therapeutic effect, depending on what you're looking at. And so this has ended up being a problem. So like, how do you get around that? How do you avoid that? So one approach was taken by a company called Sanofi Recherche, which is a French company. They were really excited about an antagonist, it was a CD1 receptor antagonist. And so they developed this drug. They were so excited about this because the idea was to use it as a weight loss. So you activate the system, you get the munchies. Block the system, you get the opposite of the munchies. You get a lowered appetite, right? So they did tests in these clinical trials. People were losing about 10 pounds, very exciting. It's actually not that much, but you can gain it all back if you stop taking the drug, which of course is the perfect drug. But so they were so excited about this that they bought a larger company to get access to a certain market, a certain world market. That would be us. That to get access to the American market, world value. That was their thing. So the French, they thought they were going to make so much money off of this. And and also there are other things. So there's actually evidence you use it as a means to promote cessation of smoking. They were finding that we didn't really figure out this part yet, but there was seeming an improvement in the blood chemistry. There was, however, one problem. This was approved in Europe. It was never approved in the U.S. So what's the opposite of euphoria?
Depression.
So despondence of people, there was an increase in the suicide rate. There was enough of an increase in the suicide rate with the antagonist that they pulled it. They felt that it was a bad move to. So because they lost a lot. I mean, they gambled, and this was a good shot. They couldn't really predict that. So people are working on variations of this approach. That's a part of the problem with the medical prospect is there are just too many targets. So you look at the brain that I showed you, the brain image, those receptors are everywhere. And so you have figured out how to cure Alzheimer's disease. All you have to do is get that receptor in there and to target a particular set of neurons. The problem is you hit everything else as well. And so you get a lot of side effects. It's a real problem with cannabis. So one thing that people have been focusing on is, well, and how about enzymes? What about the things that break down cannabis? There are things that make them in the body, because there are all these lipid enzymes that make it break down these lipid transporters. And also other ways of activating or blocking the receptors. This is what people are focusing on now, hoping to get a more selective effect. So switching gears, this is something that people often ask about. They say, well, what about heroin? Is it dangerous? And for that matter, given that this is everywhere in the brain, and given how many people smoke, why aren't people just dropping like flies? Why aren't people suffering more than they seem to be? And there have been various studies. This is just an example from the Lancet that characterizes by various means dependence and physical harm of various drugs of use. Cannabis is right here, so low on physical harm, but in terms of dependence. Khat is one that's commonly used in the part of Africa that's a stimulant. But then you have things like cocaine, so that you're kind of going up in terms of risk of harm and physical dependence. So it's fairly low as these things go.
Is caffeine on there?
I don't think so. It probably should be, right?
Yeah.
But tobacco and alcohol
But
are
alcohol is.
both more dangerous and more dependence causing.
Though I'm afraid to stop drinking coffee. I've gone through that withdrawal. So things have changed, right? So the marijuana that people are growing is stronger. It has a higher THC content than it did just 40 years ago. So in this case, the one study found 0.35% to 3.5%. And so people have been aggressively breeding these for I mean, this is why you can get all these different kinds. You know, there's Maui, I don't even know what the names are, but you know, Maui, whatever. And they are different. And so people, they'll give you, so in my home state, actually Washington State, they just changed the law. And it's kind of this weird schizophrenic thing, because you can, you can smoke it, but I don't think you can grow it. So it's illegal to kind of get it in. And of course the feds can still get you. So it's this interesting, unusual arrangement. But these plants have been changed. And the ratio of THC to cannabidiol, which is this other component, notice they used to be kind of even. They used to be very similar to each other. Cannabidiol is not a very good cannabinoid receptor agonist. It seems to do other things. So it's complicated. There are a lot of different lipid, cannabinoid components in marijuana. And those ratios have changed. And so now the cannabidiol, and there are some variations that still have a high cannabidiol level. But in a lot of them, it's really been pushed up here at the expense of this. This has been reduced. And so this has an effect on going to be the outcome when people smoke. You get a different kind of experience. But what's interesting, this is actually getting at something that we worked on, some of our work. And I think this also gets at why people aren't dying by activating all these receptors. And that is, THC is actually not very good at activating the receptor. So it's a little like if you're driving a car and you're just kind of pushing the pedal down a little bit. That's kind of what THC is like. But there are some other drugs that really kind of slam down the pedal. We'll talk about that in a minute. But so I think this is part of what's going on. THC is pretty good at getting a medium activation. It's enough to get this euphoric effect. It's not enough to really drive the system to get some of the other effects that you can get. You can get a drop in body temperature, you can get mobility. If you go high enough, you do these kinds of injections in animals, things like that. You can get some pretty disturbing effects. But yeah, so people just tend to smoke until they're high. Let's see, and then there's also I think a real differential sensitivity, something to keep in mind. I think different people respond differently to cannabinoids. So a lot of people experience euphoria, a lot of people don't. It makes them fall asleep. It's a soporific effect for many people. And so that's something to keep in mind. And also in terms of therapies, what effects you can hope for if you're doing, if you're exploring these cannabinoids for therapy. One just kind of a note is that cannabinoids, when people smoke, it tends to be unfiltered. So you tend to do you're getting a longer exposure to the cannabinoids. But I think that's a pretty minor risk, the minor enhanced risk of lung cancer. But what we're missing is really, to some extent, long-term studies. Like it's to be able to know what are the long-term consequences of this. Something else that we've been working on, we actually did some of the first work on this. I don't know if you've heard of this, the Spice, K2, these kinds of drugs, club drugs. And, so I gave this presentation, kind of a variation on this, in 2007. This was not on our radar at the time. But very soon after we started working on this. So in the last 5 years, this has exploded. This is a really big deal. And so what's happening is people have said, okay, we can do chemical synthesis. And what we can do is we can synthesize certain cannabinoids. Maybe delta-9 THC is illegal, but maybe delta-8 THC or some other of these compounds can be made. We can synthesize them, and we can just put them onto something and advertise it as incense or whatever. And so this has really taken hold. It started in Europe, and this has really taken hold in the US as well. There have been some legal movements to try and get at that. But we'll get into this in a moment. So, but these are synthetics. And they're going through the literature, they're coming up with variations, and they're just making them. And be aware that chemical synthesis is not a perfect art. So when you make something, even if you're a top-notch chemist, there are always byproducts. And so if you're not good about it, so there was the example of, I think it's MPTP. So this is an opiate. I believe, but what happened is there was this group of people that was in California. They were trying to synthesize an opiate, a very strong one, and they screwed up. They made a mistake, and they got something else that happens to be really, really good at killing certain neurons in the brain. So what you have is suddenly all these people showing up in the hospital with Parkinson's disease-like symptoms. It's kind of rigid, immovable, barely moving. And this was because it was actually an extraordinary finding in their point, but horribly tragic. But so they basically found something that would selectively kill off a certain population of neurons that end up being a very good model in an animal model. But this is because they screwed up. And so you don't know what necessarily what's in this that's been that people have been spraying on. But what's a big deal for this? This is really changing. I mean, this is kind of it's sort of a different world, different landscape. Is the potency has increased dramatically. The efficacy of these has increased. And this is where we're going back to the example of pushing on the accelerator, THC kind of gets you a little bit. These things take you to the floor. It's maximal activation. And here is why we're starting to see things like increases in emergency room admissions associated with this. And they're uncharacterized. There's been a lot of research looking into delta-9 THC and kind of related cannabinoids, but these other things, you don't know what they do, and you don't necessarily know. If they tweak the structure a little bit, they may still get the strong effect that they wanted, but it may also now activate some completely different receptor family. You don't know what you're getting. And then there's also what's in here. So in safe spots, people have looked at the content, the drug content of various packaging, and it ends up being very variable. It's often more than one drug that people are using. So as a consequence of the spike in ER admissions, in terms of trying to deal with this, it's a lot like a game of whack-a-mole. So we've made delta-9 THC illegal. Okay, it should work. Okay, somebody comes up with spice, seems to have a lot of J to age 18 minutes. So we did the work on that, showing how potent it is. So that has become illegal in some places, but the response has been, oh, we'll just go back to the books, we'll synthesize something different, now we're going to put that in. So what we've seen now is a shift to some new, different compounds. And so we could probably spend all our time just researching these new compounds as they come out and see what their effects are. This is not, it's not really what we do. We did a lot of early work that, early being like 2009, it's not that long ago, that has kind of laid the basis for changes in the legal status of the active ingredients in the spice foods. So, and yeah, so it's very challenging at a time when you're trying to deal with designer drugs, because there's more and more of them. People are, and it's not just cannabinoids. There are also bath salts, right? There are bath salts. Because it's not, there are bath salts that you wash yourself with. Yeah. But there's also, it's another designer drug. It's a variation on it, essentially a form of amphetamine that has been, people have expanded on. I think it's con. I think they've taken the active ingredient of con and they've really supercharged it. And they have these wonderful little names, right? There's one variant called Meow. That sounds great, right? But yeah, some of those end up being really bad. So that's just kind of touching on a bunch of different topics that people have shown an interest in over the years. At this point, like I said, I'd be happy to try to answer questions you may have or make some comments.
There's some research showing that if people who have a genetic predisposition to schizophrenia, if they don't get exposed to any drugs, both prescription and non-prescription, because there's a number of drugs people don't realize, one side effect can be hallucinations. If they don't get exposed to a hallucinogen, that won't trigger their first schizophrenic episode. So for example, I've studied or I've read that THC can be that trigger. And if people don't take that first hallucinogen, whether THC or a prescription drug that happens to have a side effect that can be hallucinations, they'll never have their first schizophrenic episode. Or subsequent, it may help trigger subsequent episodes. But I also read that just because of the way plants sometimes work, that how do you pronounce that? Cannabidiol?
Cannabidiol.
Cannabidiol, thank you. Cannabidiol. That actually suppresses schizophrenic episodes. In other words, it's acting. You have an activating agent, the THC, and you have a counteracting agent in the same plant, usually because they're much more normal, equal levels. And that if you want to help someone suppress their schizophrenia, you can actually have them eat something that take like hemp protein powder, which may have much higher levels of cannabidiol. So in other words, if you avoid the marijuana and instead you take the hemp seed product, you can get it in any health food store. You may actually be able to help schizophrenia rather than trigger it.
I mean, I think there's enough evidence out there at this point that that the use of marijuana can promote and development of schizophrenia. There's somebody actually in this room who's much more of an expert on this than I am, but I won't put him on the spot. But I think part of the difficulty with this is kind of comparing experiments that are done. Some experiments are done in animal models, some in human models. The conditions vary a lot, especially experiments that were done in the 70s and 80s. Those were the days to do science. My God, you could give people just a form to fill out, and you could publish them. God, I wish I could do that. But yeah, so things have gotten a lot more rigorous, especially in the cannabinoid field. Things were kind of loose for decades, and things are getting more serious. So definitely, schizophrenia is an area of great interest for the field. And certainly this idea that cannabidiol, and there are other compounds, cannabinol and other variants, they act on different targets. It's not necessarily clear. People are talking about GBR18 as a target for cannabidiol, but this is all still work in progress.
It's just amazing. The same plant that may trigger schizophrenia has a component which may actually help suppress it once if you know how to take it correctly.
Yeah, I mean, but of course it's risky to self-medicate based on limited information, which we don't have a lot of information at this point. But there's a lot of people, because people think it's herbal, let's try it out, and my mom told me this, so be careful. Okay, other questions? Sir?
There's a lot of information online these days about people using it medicinally, eating it raw, people are making concentrates of it now. I mean, are you exploring any of that stuff? What the people are actually doing? What are you getting results? Who's doing that kind of research? Anyone?
Right. I mean, so for our purposes, what we're doing, and I didn't really talk about that much of what we do. Just briefly, we do kind of a lot of basic pharmacology is what we do for practical purposes. We have a neuronal model we use to see how this different compounds activate these receptors, how the receptors work, how do the endogenous cannabinoids work. We're very interested in how those are broken down with the time courses, things like that. But then we also do practical things. We do work in intraocular pressure. We do a lot of work in the eye. Turns out that there are cannabinoid receptors on your cornea. I'm just kind of, what on earth are they doing there? And so we have evidence that if you activate those receptors, it will increase the migration rate of corneal cells. So it may actually, we're hoping that it may have some relevance for corneal wound healing if you can speed up the process in which they close over wounds. So depending on whatever system you're looking at, you may get a completely different effect on what's happening. But in terms of looking at, I guess, various aspects of different kinds of preparations that people are using, things like that, there's some research into that. But when it comes down to funding, it can be difficult to get funding for those kinds of experiments. The idea is you're aiming mainly for therapy. You want to do something that get you some kind of a treatment for some kind of disease. That's ultimately what you're aiming for. So if you're going to do those kinds of experiments, you have to package that carefully. Not a lot of people do. It's possible, and this is kind of an interesting question. Washington State, so they just legalized marijuana. Part of what they're going to do, they're going to have a tax, and they're going to assign a certain chunk of the money that they get from that to research. And it's possible that research will be more focused on these kinds of questions. I think it makes a lot of sense. Sir?
What are they going to tax and how are they going to collect it?
Well, actually, I don't know. That's a good question.
Because if you can't grow it,
you
can't sell it, but you can only possess it.
I think you must be able to sell it.
In Colorado, you can sell it. I don't know if you can grow it, but you can sell it.
But I'm not sure how you get it. So there's this black box, this mystery.
Alaska had a law that you could grow it, you could possess it, but you can't buy it and you can't sell it. There's no way to tax it.
If they go forward with that and they start funding, state agencies start funding research, will those researchers be able to accept federal funds, say from or will they not be able to?
No, I think if you're doing legitimate research, I think that there's no particular, that's an interesting question, whether Because so we have DEA oversight, and it's not fun. I'll tell you, when the DEA agents show up, they're in suits. They are very serious. Somebody's shaking his head who has to deal with this or not. So yeah, they take it very seriously. So that's actually a really good question. Whether that would be a conflict in some sense. You would probably end up having to get federal approval in order to do it, which is a long. It basically takes you a year to get that. But yeah, that's actually, I had clearly not thought that far ahead on this, partly because I'm not in Washington State, so, but it's an interesting question. Other questions? What's your?
What form do you administer the exams and what kind of process involved?
So, I mean, we do a lot of our work with synthetics, and also with the endogenous form. And plus, again, we work mostly with individual neurons. So it's a neuron that's been grown on its own. And so there, you just bath apply it. You just put cannabinoids in the bath. It can be an issue, like how do you dissolve it? Right? Because if it's a lipid. The endogenous cannabinoids work fairly well with our solutions. Sometimes you have to have an organic solvent that'll get into solution. But for endogenous is within the body, of the body. So exogenous is outside, endogenous is inside. Yeah, sorry. But so you do have to consider the fact that it's a lipid in preparing it if you're going to do an injection, for example, into an area. We mostly don't do that. We do some topical applications for intraocular pressure. So one of the nice things about working on intraocular pressure, or actually anything in the eye, is ideally you can do a topical application in the cornea and it gets in through the cornea. But in other parts, and then presumably you're just affecting the eye, not the rest of the body. But if you're doing an injection into the body, and this is actually an interesting question. So what happens if you, what is the difference between smoking and eating cannabis, marijuana? Is it the same? Does it feel the same? Would you think that it feels the same? No, not putting it into this one. The answer, I've actually, personally, I've actually never tried, but I have heard that it is very different. And that would make sense, because what happens when you smoke, there are two things. One, you get a big spike in the levels. You get a big spike immediately also from inhalation, actually, it goes very quickly. If you eat, it goes first to the liver, it gets metabolized. It actually gets converted from delta-9 THC to 11-hydroxy THC. It ends up being a different compound. And that's actually not that well studied. We've done some work on that and found that it's in our preparation it's very similar to THC, but we call it THC, but there are lots of different kinds of THC. But so that ends up having a different effect. There's a different time course effect, and it's also a different compound. So that's kind of something that people don't think about, and that might explain partly why there are these different effects, depending on mode of ingestion, things like that.
And you can't control the dose nearly as well because you don't know how you're going to digest it. When you're smoking, you can control the dose just to the exact effect you want.
I think that's probably true. That it's hard to control, depending how well you know your digestive system, your stomach, how good a friend your stomach is.
So with either smoking or eating, how is it carried through the blood? How do the cannabinoids travel through the vascular system?
Right. Well, so that's interesting about blood, because when we think about blood, we think it's basically water, right? But it's actually it's very lipophilic. There's a lot of stuff going on there in terms of carriers and that can carry lipids. So in fact, if you take measurements, there's someone also there are a lot of cannabinoid researchers here tonight. There's somebody in here who's actually probably done this, who can look at blood or things that are like blood plasma. And looking for cannabinoids, it's full of cannabinoids. The blood has a lot of different kinds of cannabinoids, endogenous cannabinoids, circulating through the system. And interestingly, so also like tear film is similar to blood plasma in that regard, it's just transparent. And then also the intraocular fluid is also like that. So there's probably transport of cannabinoids through both of those as well. I don't want to go off on that subject, but so questions?
So I read an article that was basically alleging that the whole DEA federal enforcement system was making it really difficult for a lot of research on marijuana to be done. I mean, do you know, I mean, I guess if you're working at sort of the epicenter of cannabinoid research, maybe they sort of look more favorably on your applications, but do you know, like, is it hard to get the actual drugs to do research with or get your grants approved, like more so than for other
No, I mean,
substances?
that's a good question. Yeah, so but basically, if you go through the process, it's a year-long process, it's not much fun, of getting DEA approval, then you're fine. And it's not easy to get the drugs, but yeah, they'll supply some drugs for you, so some of them you actually get for free. Some of the synthetics, things like that. Others, if you have the license for it, you can get them. So actually getting the drugs, once you have the approval, is usually not that hard, unless you're getting one of the obscure ones, in which case you can do that. But to a large extent, I mean, so this is an interesting point, that what is scheduled and what isn't. So delta-9 THC is scheduled, so that you have limitations. But 2AG that we were talking about before, that's our endogenous drug. That's mostly what I use. And so you just get that from a commercial supplier. There's no DEA involvement in that. So to a large extent, I hope that doesn't change, but to a large extent, that's actually fairly good. But it has, I can see if you're trying to do research, you're thinking, you have this great idea, but you do something completely different. You think, oh, I bet it's cannabinoids. This would cure everything already. Then you're confronted with this process. Oh, but it's going to take me a year to get approval for it. Yeah, I bet some people are discouraged and don't do things that they might otherwise. On the other hand, you can then call somebody and say, hey, this is what I've got. Can we work together? So there are ways around that.
Well, I think anytime you have IRB, so anytime you're experimenting on humans, you have different sort of approval routes you have to take. So I'm with the lab, so we don't do human work, so it's easier for us. But I can see how if you're trying to implement a protocol in humans, that will be a lot longer in terms of review process. But I do have a question. So it's kind of to play devil's advocate. So we've been talking about good side of marijuana and THC and the cannabinoid system. And an excellent point is that cannabinoids can be neuroprotective because they do decrease activation of the neurons sometimes, and you don't get excitotoxicity from certain things. Also, you can treat certain disorders like post-traumatic stress one. However, can they be oncogenic?
Well, so
If something is neuroprotective, so you're keeping a neuron around longer and it can acquire mutations. So people have been asking the question. I'm not up to date on that literature because that's not part of my research, but do you know?
No, but that brings in one really important point, which is something I didn't talk about, which is neuroprotection, right? So I showed you this model of these two neurons communicating with each other. And there's something called excitotoxicity. So when neurons fire too much, they release too much glutamate, you end up getting too much calcium coming into the cells. Calcium's bad. You need a little bit of calcium to come in and enable your signaling, but too much calcium ends up killing the cell. Excitotoxicity, glutamate excitotoxicity. And so because cannabinoids block the calcium channels, you actually end up, if you activate that system, you reduce that risk depending on what the circuit is and what the system looks like. There's a lot of interest in cannabinoids as being neuroprotective, protecting the brain using that mechanism. That's actually very kind of a big area of study for a lot of potential. But I think the second part of your question was basically, if I understand correctly, by keeping the neurons around longer, by letting them live longer, are you making them more susceptible to other things like cancers and whatnot? And I would say, we're ultimately we'll die of something. Right? So you're always at risk of something, and if you've made it past a certain age, you're at greater risk for other kinds of things to develop. But if you've managed to get that far as a result of a treatment, that's great. That's great. And then maybe we can treat the cancers that go to the origin of the cancers. But in terms of actually being oncogenic, there are other cannabinoid receptors. There's some talk of maybe GPR18, depending on how you activate it, that might be oncogenic. But it's much too early to say. But it's CB1, CB2. There's another interesting thing. So CB2, we haven't talked about at all. In the immune system, if you activate that, depending on what cells, it's involved in a lot of immune cells, it tends to suppress those cells. And so I think it's fair to say that the net effect is a suppressive effect on the immune system. But again, it probably depends on what you're looking at.
The long-term research doesn't seem to show lung cancer, like you were pointing out. in most other cancers, especially ones associated with smoking, there does appear to be a 50%, the only thing I read in the negative column, a 50% increase in aggressive testicular cancer. That's the only cancer I've heard of that it may increase. But that was only one study. I'm not saying that was a series of studies.
Yeah, that, 50%. You always wonder about what their sample sizes are in that case.
And it has to be replicated. You know how it is. That's what's only, you can find lots of outliers in individual studies.
You have to be skeptical with dealing with studies. Actually, let's sort of go to her first.
I was going to say, given the controversial nature of this type of research, what degree of interaction, if any, does your research and others generally have with government regulation and advocacy for or against this type of chemical or drug or however you want to classify it?
For the most part, not very much. I mean, I think our main job is to do the research and leave the politics to the politicians. But on the other hand, I mean, I view my role here as informing, giving people information, because there is a growing shift in the sense of what cannabinoids are, and how dangerous they are, and what role they play in society, the change in legal status in certain states. I'm fairly agnostic about this myself. And I think in that respect, I think I'm a good person to talk about it. But there aren't a lot of avenues necessarily for scientists to influence policy, apart from providing information that people then use to informing their decisions. And so an example of that was with the spice and K2 work. I think our work actually made a big difference, because that really showed that pretty concretely that these are very strong, very efficacious agonists at the receptors, and therefore potentially more risky. That answers your questions. Back to you.
I was just thinking about studies where you have where you see such drastic effects, and it's dealing with sort of hormonally mediated cancers. Whenever you smoke, you get munchies, or you use THC, you get munchies. You get probably increase in body mass, and your fats, the proportion of the body, is sort of very highly correlated with your hormonal levels, especially for proliferative hormones such as progesterone and estrogen. So it could be that people who use heavily and they have an increase in BMI, and then therefore you see an increase in, or like in metastasizing effects of cancer. So that's like always my question: how is it done? Like, is it controlled for? Because I mean, diet and sort of BMI has been ignored, like in a lot of like pharmacological studies where they match age and socioeconomic levels, but don't always match for BMI unless it's like a dietary directed study. But I mean, if it's a lipid thing and it's being carried by lipids and it's being like sort of, it can diffuse into the cell through lipid-like properties. So,
but basically what you're talking about is kind of a secondary effect then. So you have munchies, presumably if you have munchies a lot, you end up gaining weight. I don't know how much that is the case, whether people end up smoking so much. I hadn't really kind of made that. An interesting study I can see is are heavy smokers, do they tend to also be heavy in that regard? I don't know that that's really the case. But then there's this question of whether maybe that increase in BMI then might have a secondary effect with the hormonal system, things like that. It gets much more complicated, because then you're really changing a lot of different things, leptin levels and things like that. And that gets pretty hard to assess. One just kind of an aside, this reminds me of another thing we can get to your question in just a moment. This is highly speculative. It's just kind of a funny idea. But this was tossed around at the last conference I went to, which is, so these cannabinoids get stored in your fat. And so imagine you're gaining weight, smoking a lot at a particular time in your life. And then you say, oh God, I've got to change my life. I need to exercise a lot. In principle, you think that as those fat cells are dissolving and the cannabinoids are being re-released into your bloodstream, you might actually get high from exercise. I think that's unlikely, but it's kind of a funny concept. I like to think of that. I guess that's a question back there. Jonathan.
Yeah, has there been any research or studies done on neurological dependence upon cannabis, or addiction, I guess you could say?
Yeah.
I mean, does the brain become attached?
So, yes, basically it does. It's not nearly as bad. I think that's pretty safe to say at this point. It's not nearly as bad as things like cocaine. There is some evidence that you can induce withdrawal, for example. But it's harder to do. If you do with cocaine, for example, all you have to do is stop giving an animal cocaine, and and they'll start experiencing withdrawal symptoms. Coffee, for that matter, stop drinking coffee for 36 hours and start experiencing withdrawal symptoms. But it's less the case. So with animal work, for that you actually have to give the antagonist to induce withdrawals. So I think yes, but it's not as strong as others. But it's an issue, and I think again it may vary a lot from person to person, and that's hard to assess.
Hemp protein powder, surprisingly, when I was, I told you I was trying it, I was doing it actually for extra protein for weightlifting. And it was mildly addicting. I was waiting for my hit like during the day. And I'm like, I didn't feel a high off it. There was no THC content's extremely low. That's why you can sell it legally. But I was like, this is mildly addicting. So it still has hemp with high cannabin, with high that thing. Cannabidiol, thank you. Cannabidiol does have a mildly addicting. So it was just so funny to experience.
Yeah, it's hard to say what we develop an attachment to. Okay, questions? Other questions? So over here, yes.
You mentioned about effects of the immune system. In what ways does that manifest itself? That production of white blood cells?
And I should say in advance, before I get in trouble, this is not something that I've studied or looked into very much. But the last time I looked into it, it was just that there were certain cells where you look at kind of activation, like macrophages, things like that. I think that they were less active as a result of activation. But it's hard to say what that means necessarily. And also there are things like desensitization, which that's actually another issue with cannabinoids, is if we're trying to use them for therapy, how fast does this affect desensitize? Because you may have a dynamite drug, and it works for days. But then after 2 weeks, your receptors are desensitized, you're having no effect anymore, you either have to go up in the concentration, in which case where does that end? So that's another real issue
They downregulate
for
or
cannabinoids
upregulate.
in terms of therapy. So there's a lot of interest in them because they're in all these different cool places. And you think, God, there must be some way to take advantage of them. But it's pretty challenging for the cannabinoid field. Question?
So you talked a little bit earlier about how different people sometimes experience really different effects from even just like the same strain or whatever. And this could be totally speculative, but I'm wondering what you think might be sort of the underlying mechanism for that. Do you think like people just express the receptors differently, maybe like in different quantities or different parts of the brain?
I think it's very likely. There's also, this is kind of a funny thing. So one other thing we did in terms of our research was to look at species difference of how the receptor works. Because we do a lot of our work in rodents. We have these mice, these knockout mice. We insert a new gene, a modified gene, to see how that signals. Very cool technique. But we're doing basically mouse and rodent cannabinoid physiology, which is great for the mice. The mice will cheer. We're going to cure mouse cancer. But the question is, well, how does that signal relative to the human receptor? And we kind of figured it would be the same. We just kind of thought, let's just try that, see what happens. Turns out the signal's very different. So the human receptor doesn't activate as well as the mouse receptor. So that is interesting. There's a big difference too. It's really about half as much. Furthermore, there are splice variants. So not only do you have your receptor, but as it's being packaged, depending on the circumstances, you can get a different receptor. You can get part that's been snipped off, or part that's been substituted, but it's taken out from the middle, things like that. So you end up getting a different receptor that behaves differently. We've actually characterized that a little bit too. We found that one of the splice variants behaves very strongly. So depending on what's, this could be, this could be epigenetic, or it could be environmental. Something might stimulate, bring in the CB1As. This is what we need right now in this particular circuit. And so it's all kind of hard to predict what controls the sensitivity. That's a good question. You have been raising your hand a lot.
Yeah, so I was initially thought of this when you talked about the different effects to begin with. And so because a lot of research, like in social psychology and sociology, shows the way in which coming to enjoy marijuana is this process of learning. That's this process through interaction. And so it's interesting where we have these studies that are in labs and talking about mice and so on, which clearly, don't have culture, don't have, these interactions in which they learn to use this. So I guess how important is it, do you think, that this, scientific research into marijuana is so kind of disconnected from the social and actual experiences that people have with marijuana? Because I was also thinking about the idea of getting funding to do research. Well, funding to do research about marijuana could also theoretically be funding to look at how people interact with each other when they're high and this process that people go through and so on. And my thought is that type of research is research that largely can't be done. And so how do we account for this lack of social context? Or does it matter, or what do you think?
Well, no. These are kinds of questions that people even ask, and I'm glad you raised this question because as a society, society needs to think I think partly the issue now is that it's illegal. So you think, well, what's the point of studying that because I shouldn't be doing that anymore.
Exactly.
But this is changing, right? And I suspect that is going to shift further in this country. Assuming that it does, these are questions that society needs to ask. Do we want to learn about this? It probably makes sense. We learn a lot about aggression and alcohol use, right? These kinds of studies are done routinely in psychology departments, things like that. So as the legality changes, those kinds of experiments may become, probably should become much more common. It's a good question. And again, it should be part of the debate as that status changes. Question?
This is actually playing off of our original one, just kind of how people are experiencing the effects of marijuana differently. So you mentioned before about how it's processed differently when you're, eating it as opposed to smoking it. It's processed differently, does that really alter, the effects significantly? Do we know, what the actual perception would be and, how it would manifest itself?
I don't think it's been studied, yeah. I don't think, and again, it's how do you do those kinds of studies and get away with it, essentially? Back in the 70s, those were the days, right? But there were studies where you could inject people with THC. Wow, that's so cool. You just ask them, what do you see? My favorite was, I was very interested in visual effects, so my favorite was, so the highest dose, one person said that the doctor, because they were a lab coat, the doctor looked like an alabaster tortoise. Very interesting. So these kinds of studies are very difficult to do at this point, but maybe that'll change.
I was just wondering, first I wanted to say I wasn't clear on the majority of the studies on cancer in marijuana is that it lowers cancer overall. That one little study on prostate was separate. From what I've read, it appears to lower cancer overall pot. I just didn't want to get the impression that it raised cancer. Yeah, the research goes the other way. This was just one little study. What I was thinking was, why doesn't your department have on its website have people be able to talk about their unique experiences with not just marijuana, but maybe spice, K2, bath salts. That might let you keep up on if you get enough hits on this new type of spice, or it might let you keep up on what's the question to ask right now. And it would be a way for people to put these experiences on and say, here was the difference, here was the difference. You could even have as a questionnaire, was there a difference between smoking it and taking it? What was your experiential difference? And you might be able to get enough data out of this that you can get one or two really good major studies from something you didn't know until you got the user experience. There's lots of people out here willing to do field work on this.
That's right. And basically, we have conducted the largest unsupervised clinical study in history, stretching back thousands of years. We have all this information. No, I think it's a good idea to, the kinds of questions that we're asking are much more based on the endogenous system. It doesn't make a lot of sense for us, but for someone who's interested in it, I think you want to see what it's. I've heard some amazing stories, and frankly, how I got into this particular area of research was through a story of an acquaintance of mine who had a bad drug experience. Basically, I always put him to sleep, so he thought, I must not be smoking enough. So he basically was this constellation of really unfortunate effects. He ended up basically wrapped in a blanket on the floor because his body temperature dropped. And that's what happens if you give a high dose to a rodent, you get a drop in body temperature. And he also had visual effects. That's what got me thinking about vision. It was kind of like it seems like a bad TV reception, but for hours. You got to look at things. sometimes they don't work. It's all going to keep us busy for a while. Okay. Cool. Hey, I really appreciate all of you turning out. This has been great fun. So Science Cafe, so please you get the word out. That's kind of the big thing. Main thing is word of mouth. We have a website that kind of lists the information. So if you type in Science Cafe Bloomington, you'll find us for sure. Get the latest. But get the word out. We do this roughly every 2 or 4 weeks, depending on how the speakers work out. And we have a whole lineup of speakers coming up for the next couple of months. So please come. We will probably do it again here. And so we'll probably do it on Tuesday or Wednesday. So
Whose artwork is this all?
don't ask.
I heard it was yours.